This presentation will focus on our work over the last several years to improve Rhizoctonia solani resistance in table beet, which is one of our most persistent problems in the production of both the fresh market and the processing beet here. I don't have to tell this audience. We're very grateful for the support of the Midwest Food Products Association to help support this work over the years. Well, the disease is, as you know, very destructive to beet. People often will call it root rot or crown rot, and you see both of those symptoms here in this image. You can see the blackened petioles and lesions forming on the petioles at the top, the crown. And then you also see the pitted roots, and sometimes these pits can go quite deep. So even if you can peel the beet, in the processing plant, you still can suffer a lot of losses. And these lesions are really a big problem. This fungus is quite difficult to control. The openings on the roots can be quite prominent, and this is an example of what the kinds of symptoms that we can produce in our screen in the greenhouse, which is one of the experiments that I'm going to be talking about. This disease, this pathogen overwinters as sclerotia and can live in crop residue, and it's very, very difficult to predict its presence in the soil and its infectivity in any particular season in any particular year. So while fungicides can be an effective tool, it's hard to know when this pathogen is going to be at its worst. We know that there are 12 anastomosis groups for this pathogen. We are in the process of looking around the Midwest to try to understand a little bit more about its distribution. And we're also interested in the types of control methods that you all use in your beet production, such as seed treatments and various fungicides and crop rotations, to see if those can be useful. But ultimately, our program is a breeding program. And so we're interested in contributing to those tools by breeding in some resistance. And that's really the goal of this work. The work that I'm going to talk about has been done by Katarina Wigg. I think some of you know Kat. She is a PhD student in the plant breeding plant genetics program based in the Department of Horticulture. And she's developed a lot of the techniques and approaches that I'm going to talk about here. One of the things that we realized when we started working on breeding for rhizoc resistance is that you can screen the outside of the beet for symptoms. You can also cut the beet open and look at internal tissue damage. The sugar beet folks who really pioneered this work, and in fact, I should say that the group out at Fort Collins, Colorado, did the most to develop resistance to rhizoc in sugar beet, largely focused on the exterior of the beet. And they did a good job with that, but we realized that it's important for us working with the vegetable to be able to have a handle on both external and internal symptoms. And so our work combines those two pieces. The first experiment that I'll share is an experiment that looked at 94 different plant introductions. These came from the USDA National Plant Germplasm System, and they were collected all over the world. Some of them are less well-adapted table beets than you would ever grow in a production field here, but nevertheless, they represent interesting sources of genetic variation. The experiment also included 56 commercial cultivars and publicly available inbred lines that seed companies would use to make a table beet hybrid. And then we had checks in this study, which included resistant and susceptible sugar beet breeding lines. These are the ones developed at Fort Collins, along with one of our standard table beet inbreds that was developed in our program, W364. So these materials were inoculated using a particular isolate that is quite prominent in Wisconsin. And they were rated in a greenhouse after inoculation at 11 and 13 weeks after planting. And both external and internal characteristics were evaluated according to the scale that you see on the slide. And then in developing the data set, Kat used a weighted average where 25% of the focus was on the external and 75% on the internal rating. And so here's an image you can see of what the numbers might look like with the 4 and the 5. You're really getting up into some really damaged roots, whereas a 2 could potentially be peeled or cut away a little bit to get some useful tissue. Just showing you a couple of lines from the analysis of variance here. The effect of accession in the PI study was very significant. There are huge differences among the plant introductions as there are among the cultivars, and a very significant difference in evaluation time. That is, beets perform differently at 11 weeks after inoculation than they do at 13 weeks. And we were particularly interested in the progression of the disease. I'll talk about that in just a moment, but here is a chart that shows you the variation that we found in the plant introduction screen. And so one of the interesting things that you see about this is that some of the plant introductions down at the lower left hand of these are coded, but some of these accessions exhibit a very significant amount of resistance. Now, again, these are not materials that you would likely be growing as a cultivar, but they represent useful genetic variation that we can cross into our breeding material. So this is actually quite promising, particularly the ones that are in green. And I'll explain those in just a moment, but the green bars give a kind of response to Rhizoctonia that's actually quite useful for us. This is a chart of the cultivar and breeding line version of the experiment. And you'll see also a lot of variation present here as well among cultivars. Fortunately, there are some cultivars and particularly some breeding lines that exhibit a fair amount of resistance that can be used in our breeding program. And so this is encouraging. Kat used spline regression, which examined the area under the disease progress curve to look at the slopes. That is the period of time from inoculation to the first evaluation at 11 weeks, and then in between the two evaluation periods. And she assigned 4 different categories to those slopes. So they are a slow disease followed by a spike, a quick, meaning a lot, early disease followed by a plateau, early disease followed by a constant high level of disease, and a slow constant. These 4 characteristic kinds of slopes were useful to us in discriminating among the germplasm in these studies. And I'll show you what I mean in just a moment, but I'll first say that there was lots of variation present. We found several accessions, both in the PI screen and in the cultivar screen, that performed just as well as the resistant sugar beets. And some of the best resistance that we found had the slope of what we call the slow constant. That is, they appear to have a lower amount of disease to begin with, and that disease does not progress in the tissue. The ones that were more diseased and were more susceptible to Rhizoc had all the other 3 disease progressions. And so the slow constant is something that we can then begin screening for in our breeding work. And I'll just show you a few examples here. These are this is the PI screen. I'm just showing you the top 5, the worst 5 and the best 5 at 11 and 13 weeks in this particular slide. And you can see these PIs. Some of these are old varieties or materials that came from other parts of the world, including India and Asia. Some of them are quite resistant, and some of them are quite susceptible, and there doesn't seem to be any geographic pattern to them. We just can choose materials that look like they respond well. And so, for example, this Ames 22-164, this Ames 22-164 is an ugly cultivar that you probably wouldn't grow, but nevertheless outperforms the resistant sugar beet by a significant amount. So that's one that we can use in our breeding program. And in the cultivar screen, We also found quite a lot of differences. One of the interesting things that we found is that something like Ruby Queen, which you might use in your production fields, or even Red Cloud, these tended to be quite resistant at 11 weeks. And if you look at the chart on the lower left, they would be among the top 5 cultivars at 11 weeks, but they drop out of the picture at 13 weeks. And so that suggests to us that their disease progression is worse than some of the other materials that we have. So it's interesting. We've learned that when you screen can be quite important. The 2 bars on the left of this chart show what we call the slow constant response. And that is that the lower disease percentages down there, they're in the 20s, let's say, those tended to be ones that exhibit either PIs or cultivars that exhibited a slow constant response. And so in terms of breeding, what I'm able to do now is look for materials in our breeding populations that have have this kind of slope in when we look at the area under the disease progress curve. And so I'm encouraged that there is some of this material present in the collection. I want to share a little information about another approach that we're using to try to understand how we can improve Rhizoc resistance. And that is called a direct challenge assay. And I have to thank Amanda Gevins for helping us learn about this type of an approach. Here, you're seeing a Petri plate in the laboratory where the fungus is being grown. And we actually grow this fungus we're growing it on a plate here, but it's stored with and grown on barley grains, and so those are barley grains there in the middle of the fungus, and you see the mat there that the fungus is growing on. And here you're looking at a slice of beet, obviously from a particular plant introduction or cultivar that we're testing, and you'll notice that it has a cube there in the center, and you'll see the growth of the mycelium from the fungus. And essentially what we're doing here is inoculating the beet directly, the slice directly, and we're measuring how much mycelial growth you get on this particular slice. So we're interested here not so much in the disease penetrating the exterior of the root, but how well the root or the sliced root supports the growth of the fungus. So this direct challenge assay gives us a chance to see how far that mycelium can grow around from the center of the tissue. And so you will see some that support a lot of mycelial growth and others that support less. And so this is another way to understand something about how the beet responds to the fungus. And so assays like this have been conducted by CAT over the last couple of years. Here you're seeing a number of different slices that are being measured with the mycelium. And in this chart, you can actually see the resistant sugar beet down at the lower left. And the y-axis here is the hyphal coverage of the beet slice. So it's essentially how much of that slice, how much of its surface area is covered by the fungus. And you'll notice that something like W364B, which is one of our standard inbred lines, is not that different, although it has significantly more fungus infection and coverage than the resistant sugar beet, but it's close. And there are several other lines from the Wisconsin program that are fairly close to the resistant sugar beet. And you can compare that to the susceptible sugar beet on the far right of the slide. And interestingly, I'll point out that Shiraz, which is a cultivar that was developed fairly recently, has been promoted as a cultivar that has some tolerance to Rhizoc. And we have found it to be not very resistant in our studies that we've done. In fact, it maps fairly close to where the susceptible sugar beet does. So this is another way for us to get some information about how beet responds to the pathogen. We're in the process of, CAT is in the process of analyzing some of the data. But in this slide, you're looking at, again, the direct challenge assay, but with a color coding system that tells you how similar the results are from the greenhouse screen, where you're inoculating the whole plant, to the laboratory where you're doing the direct challenge. And so there is some correspondence between the two, but they seem to be testing two slightly different things. Generally speaking, though, the resistant sugar beet and the susceptible sugar beet map fairly similarly, whether you're doing the direct challenge assay or the greenhouse screen. And so we're trying to look at it from both of these angles to get a more complete picture of what we think has potential for resistance, and by virtue of that, potential inbreeding. The third component of the work that CAT is involved in is to find genes associated with resistance and susceptibility in beet. And if we can identify those, we can use molecular markers near those genes to aid in our selection. And this is a huge advantage in breeding because the screens are very time consuming and they take a lot of space and a lot of time. So using a molecular marker can be an enormous advantage. To develop this part of the project, CAT crossed resistant by susceptible parents, and not only table beet, but sugar beet as well. And that is crosses between, say, a resistant sugar beet and a susceptible table beet, and vice versa. And over a period of 3 years, then generated the segregating populations that she then tested. So these were tested in the F3 generation in replicated trials. And then she sequenced DNA from each family, allowing us to then examine these populations to find markers associated with resistance. And that analysis is going on right now. And next year, I'll certainly be able to share with you if we've been successful in identifying markers. But this slide shows you the genetic variation in those populations displayed as a principal component kind of analysis. We have two populations depicted in this slide. One of them is one of our inbred lines, W357, crossed with the resistant sugar beet, and the other one is W364 crossed with a susceptible sugar beet. And so we hope that by analyzing the DNA sequence information and the phenotypes, we'll be able to find some markers associated with resistance and be more efficient in bringing those resistance genes into table beet. And some of those resistance genes are coming from some of those PIs that I showed you earlier, including that PI from Ames that looks incredibly resistant in our screen. I want to just take another minute or two before I finish and share with you a related beet disease issue. This is not supported by the Midwest Food Products Association, but is related to the overall improvement of disease resistance in table beet that our lab is involved in. In addition to the Rhizoc work, we've been part of a nationwide team looking at bacterial leaf spot, which is a relatively new disease problem for table beet and Swiss chard. And what's unique about this disease problem is that it appears to be affecting not just the crop, the processing crop itself, but also seed production in the Pacific Northwest. And here we're talking about Pseudomonas syringae, in particular the pathovar optata. This seems to be an emerging problem on beet seed production. Because of that, beet seed is coming into places like New York and Wisconsin with bacterial leaf spot on it. In 2017, and since 2017, there has been a pretty high prevalence of bacterial leaf spot in New York and in many parts of the Western United States. Carolee Bull is a plant pathologist at Penn State, and she's leading a very large team. Our lab is a part of it, funded by a USDA SCRI grant to look for an integrated approach to manage bacterial leaf spot across a number of different vegetable crop systems, including beet and chard. And so we're screening in this particular project for resistance, just like we are with the Rhizoc. We're also looking at the important aspect of the beet plant, which is the reproductive phase. So this is a picture of a beet leaf from our greenhouse where Emily Galke, who's one of our students doing this work, has been able to replicate and imitate the symptoms of the bacterial leaf spot that we're seeing out in the field. This can be done in the greenhouse using an inoculation. And then she's testing not only the vegetative growth of the beet plant and the beet root, but also the beet roots going to seed. And so she's just been planting this week a fairly large experiment to look at the bacterial leaf spot during the seed production phase. So to conclude, I'll say that our lab is hopefully making some progress to improve resistance to Rhizoctonia, bacterial leaf spot, and the third disease that our lab works on, which is Cercospora. These are two fungal and one bacterial disease that are really important in the beet crop. And we hope that by doing this screening, doing this breeding work, we can help produce some varieties that will have tolerance or resistance to all three of these important diseases. Thanks everybody for your attention.
Thanks very much, Erwin, for your presentation and work. We have a question for you. On the mycelium growth assay, have you confirmed whether the growth on the beet slice is Rhizoctonia or another saprophytic fungi?
We can, thank you for that. Thank you for that good question. We have been able to confirm by looking at the hyphae that this is Rhizoc. So we're really careful in the way we inoculate, but we've had to then confirm that indeed we're looking at the spread of mycelium from the Rhizoc, from Rhizoctonia solani. So thanks for that good question.